the next speaker. It's Lisa Elliott from Australia, from Biologics, and Lisa will talk about bacteriophages in aquaculture and what can we expect from bacteriophages for this industry. So Lisa.
Thank you. Just forward and back. Okay. Yep. Okay, thank you everyone for your attention today, and hopefully I can educate you a little bit on bacteriophage, or in Australia we call it bacteriophage, so I apologise in advance for slopping between phage and phage. Okay, so we've heard a lot today about how fast aquaculture is growing. It's one of the fastest growing food production industries in the world. And since the last 30 years, it's grown substantially. And we've seen a lot of that being discussed today. It's expanded. It's expanded across so many countries now. It's diversified and intensified. For people that don't know what this actually is, this is Camao in Vietnam. And every one of those little tiny squares there is a shrimp pond. So when I say it's intensified, it's actually quite extraordinary to look at. It's diversified, and as we heard a lot this morning about all of the different fish and plants and molluscs and crustacea, it gets more and more diverse as we get more and more educated. Even though there's been a lot of research into aquaculture, not as much as some of the other industries that we've heard about this morning, there are still a number of threats. The major ones, from my perspective anyway, are chemical residues and infectious disease. The major chemical residues that we see are sometimes from effluent, sometimes from agricultural sprays and products, and particularly from antibiotics, which is becoming more and more of a problem as the years go by. The infectious diseases, the three main components, although parasites are a big part of this, are essentially viral, fungal and bacterial. And they're the ones that we tend to focus on the most, although parasites do come into that situation as well. So how do we control infectious disease? We've come a long way in the education and the experimentation and the development of things such as vaccines. And they've become a mainstay for a lot of the industries that we work in. We've also come a really long way in sustainable management practices. Again, making it really allowing us to grow very quickly. And from my perspective and from my background, it's all about a healthy microbial community. Balancing that system is crucial, or otherwise we have all sorts of problems. When we put antibiotics in, when we put chemical treatments in, if we've got bad management practices, it goes bad very, very quickly. And when this happens and it all hits the fan, it's really hard to get it back on track. So we try not to let that happen. I'd like to talk a little more now about antibiotics, because this is an area that I spend a lot of time working in. We tend to put our antibiotics in aquaculture a lot in our feeds in order to control some of the disease issues. And as we've heard this morning, there's a rapidly spreading increase in microbial resistance and a rapidly decreasing amount of antibiotics that are being developed and approved for us to use. Antibiotic resistance is a shared responsibility. We heard a lot about that this morning as well. It's not just aquaculture, it's not just agriculture, it's other crops, industrial and household chemicals, and human healthcare settings and community and treatments, etc. And it's up to us as a whole group to be able to look at this responsibly. Regulations and restrictions are now a major pain in the ear. And it's becoming more and more difficult, and some countries are banning a lot of the antibiotics that we typically use, so it's becoming more and more difficult for us to control bacterial disease in particular. So we have to think of other ways of dealing with some of these issues. So bacteriophage, what is bacteriophage? It's been around for a very long time, but in my experience, most people either don't really know what it is or are quite frightened of it. So let's talk a little bit about it. What I'd really like to talk today about is it safe? What's the application? Can we use it for anything in aquaculture? And is it sustainable? So let's first have a look at what bacteriophage actually is. It is a virus. And a virus is a small infectious agent that replicates inside living cells of other organisms. How does bacteriophage differ from a plant virus or an animal virus? It is specific for bacteria. It does not have the mechanisms to infect other eukaryotic cells. That's not its job. It's been around and researched since the very early 1900s. And these two gentlemen here, Frederick Twort and Felix de Herel, and I apologise again for my accent, were the first two gentlemen to start describing bacteriophage back in the early 1920s. And the medical uses, the historical medical uses for phage are actually quite diverse and have been around for a very long time. In fact, the Red Army Soldiers Kit included bacteriophage preparations to combat dysentery and gangrene and other such things. So it has actually been used and trialled in a number of places for a number of years. Where are they found? They're the most abundant entity on Earth. So 10 to the 32 phage exist on Earth. That's the number that some statisticians come up with. They're everywhere. They're in the soil, they're in the water, they're in the air we breathe, the top of the table, the breakfast you ate this morning, the ocean, absolutely everywhere. Wherever there's bacteria, phage will be present. In every single drop of oceanic water, there is 1 million phage particles. So that just gives you an idea of how much is out there already in the environment. How do they work? There's two essential life cycles that we see with phage, and this is where people, I think, get a little bit confused as to whether they're beneficial or they're dangerous, or, you know, can they mutate and we all grow flippers or something like that. So the lytic cycle is the cycle that we prefer for phage therapy. But I'll go through the lysogenic cycle first. So the first thing that a phage does is it has to attach to the outside of the bacteria. It's extremely specific. So the receptors on the bacteria and the receptors on the phage have to match like a lock and key. If they don't match, the phage won't attach and won't infect. So they can be extremely specific or a little bit more broad spectrum. Once it's attached, it introduces its genetic material into that bacterial cell. Then it has a choice. It's either lysogenic, or it either goes into the lysogenic cycle or goes into the lytic cycle. Now, if it's purely a lytic, if it's purely a virulent phage, it will only go into the lytic cycle. But if it's a lysogenic or a virulent temperate phage, it can go into the lysogenic cycle. So the genetic material is in there. If it goes into the lysogenic cycle, that bit of genetic material is integrated into the host chromosome and becomes what we call a prophage. The bacteria then divides as it normally would, and that little bit of that prophage is in each of those daughter cells. If the bacteria is under stressful conditions, for whatever reason that could happen, it then initiates the lytic cycle. So the lytic cycle, the bacteria then transcribes the phage DNA. So the chromosomes are all broken up and everything starts. The replication of phage DNA and proteins is initiated. So the bacteria is no longer alive. It will not make any more bacterial proteins. It only makes phage proteins at this stage. The phage are assembled, and then the cell is lysed. So there is no genetic transfer here. The cell is lysed. So what does that look like in the real world? If we have a look here, and I'll probably press the wrong button. Oh, and I did. Sorry. Sorry guys, no glasses on.
One moment.
Okay.
Yeah, but we just need to... Yep.
Thank you. Thank you. I'm not touching that again. So in the absolute top corner, you can see the outline of a bacteria. That's actually Aeromonas hydrophila, and it was a major disease issue in a freshwater crayfish farm in Australia. And then to the side of that, you can see sort of an outline of something. That's a lysed bacterial cell. And from that, all of those very, very small circles are phage. So from one phage that initiated the lytic cycle, hundreds of phage are produced. This can take as little as 15 minutes for this process to happen. From initiation to lysis is very quick, depending on the bacteria that we're dealing with. So again, two cycles, lytic cycle and lysogenic cycle. The lysogenic cycle is really important in molecular biology and has been used for a long time. It's used for phage display and to produce antibodies. It's used for vaccines, and it's used as a gene transfer system. So it's actually very, very useful in the molecular world and for looking at genes. However, it is not suitable for phage therapy because it does transfer genes around, and that's not what we want in our system. The lytic cycle, however, is suitable for phage therapy because it destroys the cell. At the end of its cycle, the cell is destroyed, and that's what we're looking for. So again, let's come back to safety, application and sustainability. Phage therapy is essentially the use of phage to control specific pathogens or problematic bacteria. That's what it is. It's very simple. There's a paper from this group that's a recent paper, 2012, and they describe the use of phage therapy in aquaculture very, very well. And I would encourage you, if you're interested and you want to read a little more about this in much more detail than what I've just said, please try and get this paper and have a good look at it because it's actually really well written. And in this paper, the authors have said that phage therapy should very, very much be investigated as a measure of control for pathogenic bacteria in aquaculture because the results and the information they gained was really quite promising. I'm here on behalf of Biologics, and in the past 6 years, these are some of the projects that I've been working on with Biologics. The very first project we did back in 2008 was looking for a phage therapy for the control of luminescent vibriosis in shrimp hatcheries. We had some really good results with that. Then we did some work with Streptococcus agalactiae in South America because that was a major problem back then. Again, it worked quite well, but these are all very small trials. More recently, just last year, we had a major disease outbreak in a crayfish hatchery, and it was Aeromonas hydrophila, and we developed a really good phage therapy, and it worked extremely well, and I'll talk a little bit about that in a minute. And also, more recently, we've had a look at Vibrio parahemolyticus that's associated with APANS or EMS. I'm not an expert in this because we don't have the disease in Australia, so I have to go across countries, and I'm sure there's other people here that have got far more knowledge on this than I do, but I can talk to you about the phage therapy based on that Vibrio parahemolyticus. So just as a case study, just to show you how we actually get this into an aquaculture system, this was the development of a phage therapy, as I said, for Aeromonas hydrophila. This particular farmer had a hatchery for Charax quadricarinatus, which is an Australian freshwater crayfish. He was experiencing greater than 90% mortalities in his stage 2 larvae. So essentially he was going broke because you can't manage with 90%. They tried probiotics, antibiotics were tried. Within 2 weeks, the antibiotics were producing resistant Aeromonas. They'd tried many, many things over 2 years, and essentially it was all unsuccessful. They were not able to control this pathogen. So the farmer asked if we could produce something that would control this pathogen for him in his system that wasn't antibiotic. So we said, yes, we'll try our phage therapy. We did isolate Aeromonas hydrophila, and it's a major global problem, Aeromonas hydrophila, but one of the problems is it's also a common environmental bacteria, so you have to make sure that you're actually knocking out the right bacteria. We did develop a phage therapy, and there were several different phage that went into this cocktail, and this is a a photograph down the microscope showing the dark area in the background is a lawn of Aeromonas hydrophila, the pathogen. Those light areas, the little circles that you can see, is cellular destruction caused by the phage, or plaques as we call them. So if we have a closer look at what's going on, there's 2 phage there from the cocktail. There's this large one in the middle here with his little boots on. He's ready for dancing, that one. And right up the top there, which is not particularly evident, is a smaller one. So phage come in a number of different sizes and shapes and families, and that's just an indication of 2 of the phage that were in that particular cocktail. And this is what we're seeing, again, where that cellular destruction is happening and those plaques are being developed, is where the phage is infecting the bacteria and the bacteria is being lysed. So this is just a shot of the incubators. So we had less than 4 weeks to produce a phage preparation to help this farmer, or he was going to have to go and cut cane and not be a aquaculture farmer anymore. He used the preparation daily for 3 months, and we saw absolutely no resistance over that time. This is a very small closed system, so if we were going to see resistance, this would have been the time. And we had a consistent reduction in the pathogen over that time. So he was able to produce everything he needed. He had greater than 90% survival across that time. He also had a reduction in the biofilm that was causing a lot of problems in his system, and he produced very healthy little craylings that went off to feed the masses. We've also been, as I said, looking at the control of Vibrio parahemolyticus in Appens, or EMS. I'm sure you all know about EMS, but it was first described in China in 2009, and then over the past 4 years, it's ripped through Southeast Asia, causing mass mortalities, huge economic problems, and we still, to this day, haven't really, as far as I know, got a solution. So one of the things I was asked, can we actually find phage against this pathogen? Essentially, this is just the mortalities in some of the farms, some of the background, but I'm sure you all know this, so I'll just rip through because I'm getting the finger. So what we did was we were able to work with some groups in Vietnam. They were very, very kind to offer us some help. As I said, we don't have this in Australia, so we can't have the organism there. We developed 15 different bacteriophage and treated a number of different isolates. There was over 114 Vibrio parahemolyticus that were isolated from various regions over various times from various places, including moribund shrimp and water. And what we saw was a 80% reduction in the optical density or biomass of the Vibrio parahemolyticus when challenged with the phage cocktail. And that is really quite exciting for us, and we're hoping to do some more work along these lines. So, again, the safety. Phage will not go across animal cells, so there's no worry about that. Horizontal transfer of genes is a major, major concern for everybody when we talk about phage therapy. It's crucial that the phage that are selected are virulent phage only and not temperate phage. They will destroy the cell, there is no gene transfer. In fact, in nature, there's up to 10 to the 23 infections per second, so it happens in nature anyway. We also select our host bacteria that have no undesirable genes, no genes that could transfer toxins, no genes that can transfer antibiotic resistant, that's all checked. Toxicology is done and dusted. It's been looked at for over 90 years, and there's been no significant side effects for human, animal, or environment over that time. The application, we can put it in liquid, hatchery, live feeds, disinfectant, powders, feed, tablets, put it on wounds. Injection, it's used in other countries in hospitals as vaccine productions and also as intravenous treatments. This is a couple of products that the Listex product is FDA approved and has GRAS status in the US and also in Australia to put onto ready-to-eat meals such as cheese and raw meats. So that's, from a regulatory perspective, that makes it a little more interesting. The sustainability. They're the most abundant entity on Earth. We can always find a new phage for a bacteria. If there's mutations or something new comes up, there's always a way for us to do that. They're nature's way of controlling bacterial populations. That's their job. Phage will mutate to overcome resistant bacteria, and they don't have a problem with being used in combination with an antibiotic or something like that. There's no problems there. The appropriate selection of the phage and the bacteria, the host bacteria, is crucial to a successful product. So just to wind it all up now, in conclusion, I believe that bacteriophage can be a friend for us in aquaculture. We've shown that they're safe, and many of the perceived disadvantages that have happened over the last several years can be addressed. Many forms of application, and they are sustainable. They are a natural, sustainable, antimicrobial, non-toxic, no threat to plant, animal or environment. And that's it. Thank you very, very much for your time.
Thank you, Lisa.
Sorry about that.
Thank you very much, Lisa. We will have time for one quick question from the audience. Look. No. Suddenly there are three. I will choose gentleman in back. We will have more time then on the discussion afterwards.
Sure.
But
Congratulations for your very interesting presentation.
Thank you.
I'm not an expert in phages and in aquaculture either, so the question is more of the microbiological nature. And I will also get from the first interesting also talk that we heard about quorum sensing. Would you expect that any resistance to phage would develop through modification of the bacterial surface in the way that it blocks the phage from attaching to the bacteria?
Yes.
And then this quorum sensing, again, have you got any indications? Is it being investigated, researched at this time?
Yes. So bacteria do become resistant to phage, so they will produce receptors that block phage. The way we get around that for phage therapy is to develop a cocktail of phage that bind to different receptors on the outside of the bacteria. So it's very difficult for a bacteria to be able to modify several different receptors without losing either its virulence or its will to live. So it's not a good practice in phage therapy to use a single phage. You use a series or a cocktail of phage together, and then we see, as I said with the crayfish, after 3 months of every day exactly the same cocktail, no resistance developed. And that's one of the things we wanted to try to see, is this actually true? Would this be a fair thing? And it is. And I think the crucial thing is in the development to choose the correct phage for the job.
That's great, thank you.
Yeah, thank you.
thank you very much. We will have, as I said, more time afterwards.